I never know that if it's recording or not, so I always get worried. So I want to tell you about transpiration. Hopefully this won't take too long because you're going to need some time to talk in your lab groups. So for transpiration, the question that we're going to be attempting to solve is how transpiration links metabolic pathways to abiotic factors. And obviously, feel free to pause this however often is necessary. So what we need to refresh ourselves on is what water potential is. And water potential is a way of talking about water concentration when we can't really calculate concentrations. Everything moves, and they move always from a high to a low down a gradient. We used concentration gradients before in order to make things move. And we've dealt with pressure gradients, but when we deal with water, we have to talk in terms of water potential. Water potential is an equation that we use in order to figure out where's the high and where's the low. The reason why we want to do this, hopefully you can see the mouse, is we're going to try and move water from the soil to the leaves, because photosynthesis is going to be occurring in the leaves. It's not going to be occurring in the soil. Down here is where you're going to have the water. So the plants need to figure out a mechanism to move water from soil up to the leaves. And that process of moving water through the plant from soil to leaves is what we call transpiration. In one of the talks that happened first semester, you were introduced to tissues that happen to exist inside of plants. And these turn out to be for vascular plants, so not all plants turn out to have this. But there are some tissues that we call xylem and phloem. So these cells here where it says xylem, X-Y-Z-L-E-M, xylem is basically a whole bunch of dead cells. There are a whole bunch of cell walls. And they actually need to function that way because we need to be able to move water through these tubes. They kind of look like little straws. And we'll use those tubes from the roots all the way up until we reach the leaves. That's our goal. So if I look at this picture, the soil has a water potential of negative 0.3 megapascals, MPa, megapascals. Remember that water potential is always a negative number. Unless we're dealing with something really squishing on it, then you'll have a positive water potential. The atmosphere is around negative 100 megapascals. So clearly, water will move from soil into the air. We call that evaporation, or the soil's drying out. But we try and manipulate it. The way we are going to try and manipulate it is make it move through the tree, make it move through the plant. The equation, or a more full equation for water potential, is all of this. So you have capillary action, you have solutes causing things, we have pressure, and we have gravity that we need to play with. We, in this class, just deal with solute potential and pressure potential. We ignore capillaries and we ignore gravity. And it's really simple math. We've done these problems before. So how are we going to make this happen? Step number one is we need to get water into the roots. And the way roots do this is through a mechanism that we call cation exchange. But roots can do is they can take CO2, which can easily diffuse in because it's nonpolar things can move right through a cell membrane. You combine CO2 with water, you make carbonic acid, which will decompose to bicarbonate ion and a proton. That proton, what we can do is I can exchange that for what exists in the soil. So if I take two protons, I can exchange that for a calcium ion, or a magnesium ion, or a copper ion. And by doing that, yay, I'm actually now absorbing things that the plant needs. But what I'm also doing is I'm adding large particles that aren't normally there into the root. And by adding these particles into the root, what I'm going to do is lower the water potential of the root. And if I can lower the water potential of the root, we'll be able to move water molecules from the water found in the soil into the root, because water will move from high water potential to low water potential. The way plants do this, the easiest way is what we call a cation exchange. We exchange a positive ion for a positive ion. A lot of plants also store water in roots, or not water, they'll store metabolites, so things like sugars in the roots, and that also will lower the water potential of the roots and allow water to flow on in. Now what we need to do is, if we have water inside of the roots, now we need to move it from the root up to a 300-foot tall tree. So the way we're going to do that is what we call capillary action. Water turns out to have four emergent properties, or among four, you can make a bigger list. So it's less dense as a solid than it is as a liquid, so ice will float. It turns out to be excellent at absorbing heat. So it has a high specific heat capacity. Water has a high surface tension, and that's because it can have this thing that we call adhesion and cohesion. Water has the ability to make 4 hydrogen bonds. Hydrogen bonds are attractions between positive and negative things. And if I look at a water molecule, these 2 little round parts, the ears of the Mickey Mouse, those there turn to have a slight positive charge, and the face of the Mickey Mouse of the water molecule has a negative charge. So what you can get is a slight attraction between the negative and the positive. If that's happening between water molecules, we call that cohesion, because it's sticking to itself. If you get an attraction between water molecules and the cell wall, which will have polar things, so it can make hydrogen bonds, we call that adhesion. And what you can get is this chain of water molecules that are all stuck to each other, but they're also sticking to the cell walls. So what we now have is we have this chain where they're all connected, they're touching the walls and they're touching themselves. And if I have water rushing in from the roots, that water rushing into the roots is going to shove this chain up. It's what we call root pressure. And then if I have something at the top that's pulling upwards, I can have the pressure from the roots shoving this column of water up, and from the leaves or somewhere at the top, we can pull the water up. And what we get is this chain in an insanely thin tube called xylem. And by doing this, it is capable of moving water from roots found 10 feet underground to the leaves 300 feet in the air. And that's the process of transpiration. So here's just a different way to look at it. When we look at xylem, it's all dead tissue, and we use this just to move water and minerals like those cations we brought in. It grabs a hold and it just pulls itself up. You might remember that there was another tissue called phloem. Phloem actually is a little odd. It deals with things that are just produced. Sorry, yawn. Hopefully you all didn't yawn at that one. And we use a different mechanism to bring stuff down. If I actually want to move stuff down, we just use gravity, which is really simple. It's called bulk flow. So the goal is eventually to get to a leaf. This is a stereotypical angiosperm leaf, meaning the leaf of a plant that makes a flower. If you were dealing with something like a gymnosperm, which is one of the cone-bearing ones, like the trees that we have outside of L4, those ones have a totally different anatomy than this. But what you'll notice is we'll have this vascular bundle, so all these tubes. These are going to be xylem and phloem, so water's going to be flowing in through these. Then we have this big old set of space where it says airspace. So we have all of this space in here that we can fill up with water, or at least with a sheet of water. And the water is going to stick to stuff. So it's not going to be filled with water. It'll have air. But what you'll get instead is a very, very, very thin layer of water all over this. It just dawned on me I should have had a picture that shows that. So I'll update it. I'll update the presentation that's going to be online for you. And I'll have a picture showing you that watery layer. So water will flow up the plant in through the vascular tissue. It will diffuse out because it's going to follow osmosis. We'll go from higher water potential to lower water potential. Inside of the leaf, you're going to have water vapor. So obviously it's going to be sucking water out. And the result is we'll get a thin layer of water everywhere. The reason for that is water likes to stick to itself. We'll have that thin layer of water spreading everywhere. With that thin layer of water, we've increased its surface area. If I increase its surface area, I can increase whatever it does. Among those things that we can do is we can evaporate water. Where will it evaporate to? It'll evaporate into these air spaces. The result is it becomes insanely humid. Obviously, some of that water that's coating everything can get absorbed by the cells, and then they can go through photosynthesis by taking the water, ripping it apart, making oxygen, and producing some electrons that we could shove through the electron or through the light-dependent reactions. But we have all this water in here, in this space. The problem is, I need to move this water out so I can keep this chain going, so I can keep pulling up more water and pulling up the nutrients that come with it. So to do that, we need an outlet. And we call those things a stomata, or stomata plural, stoma is one. Stome means mouth, and when you look at it, it kind of looks like a mouth. There's some tulips, and there's the opening. The stoma, or a whole bunch of stomata, turn out to be nothing but, and it's an opening, it's a hole. It's not an actual thing. The way it turns out to be regulated is with two cells that we call guard cells. So these two guard cells have a very simple job. They can either absorb water, and when they do that, the cells swell up, and when they swell up, they push apart, and you get this opening. Or if they let all the water out, so for whatever reason water is pumped out or water leaves due to osmosis, the cells will get smaller, they'll shrink, and when they do, they collapse and will close the stoma. So we have a very interesting mechanism just by manipulating where water is that we can open and close stoma. I'm not telling you the entire story. There's actually a lot of stuff and it deals with potassium ions and action potentials, and it's unbelievably fascinating. If you can change water potentials, you can immediately warp this, but we don't care. What we have is an opening and close valve. If we have an opening, what we can get is that water that's floating inside of this airspace can leave, which means I can pull this chain up. What I mean by pull this chain up, I can pull from the top, which means I can let more water shove in from the bottom. So we get that root pressure shoving in from the bottom. It's going to push up from the bottom, pushing water up through the xylem. It's all being held together by hydrogen bonds, which will end up moving up, however tall the plant is, from the xylem into these air spaces inside of the leaves. Inside of there, the photosynthetic cells are going to absorb water, but also water is going to evaporate, and it can leave through the hole that we call the stoma. That's the entire process of transpiration. It's moving water from roots to the atmosphere, which is what we're trying to do. Here becomes the problem, though. It takes energy to make this process happen. This isn't a free deal. Opening and closing stomata takes energy. In order to have cation exchanges, we need to have energy. The problem then becomes, what if I'm running through this process, but the atmosphere, yay, the bell just rang, is so dry that it's actually pulling up water quicker than the plant can utilize it. Meaning, the plant is absorbing as much water as it can as running through transpiration, but the water is being lost quicker than the plant can use it. It's using up more energy, it's not getting the water it should have, and it's not getting its bang for the buck. It's not being able to keep up with photosynthesis. This is going to turn out to be lethal for the plant. It's what we call transpirational loss, and it's kind of a big deal because the more loss you have, the more water you waste because the plant's not using it, the more energy this plant is using up, and the result is going to be eventually you'll kill the plant. Conveniently, plants have figured out a way around this, and I've shown it to you before, and that is photosynthesis. I've actually shown you this very picture before. What we have here on the left is sugarcane, which is what we call a C4 plant. It's called a C4 plant because when it takes CO2 and it shoves it onto something, that process there makes a 4-carbon compound, and that's why we call this a C4 compound during carbon fixation. The reason why it would do this is because this area here, which is tropical, has a lot of oxygen. Obviously, everyone's going to have a lot of oxygen. And rubisco, the enzyme that adds CO2 onto RUBP, ribulose 1,5-bisphosphate, in order to make our 3-carbon sugars, the glyceraldehyde 3-phosphates, rubisco doesn't know what it's doing. It can't tell the difference between CO2 and O2, and the result is it's going to add O2 far more often than it needs to, and it's lethal to the plant. So plants have an alternative. What they'll do is they'll fix carbon in a different cell, in what we call a mesophyll cell. So mesophyll is this outer layer. And what they'll do is they'll add CO2 onto something else, which is phosphoenol pyruvate, PEP, and it's going to form an organic acid. And right off the top of my head, I'm totally drawing a blank on it. It's going to bug me till I remember. So, oh well, it is what it is. But it makes oxaloacetate. So it's going to make oxaloacetate, which we can then ship off into another cell that we call a bundle sheath. The bundle sheath is a whole bunch of cells. This picture doesn't have it, but there's cells that line around the vascular tissue. So bundle sheath, it's around the bundle, and it makes a sheath, which is a covering. So what we can do is fix CO2 in one spot and run through the Calvin cycle in another. Calvin cycle is where we have to worry about Rubisco, but we're removing the oxygen worry from Rubisco, which is hooray. There's another issue that can happen, and that is, what if by letting this happen, absorbing the CO2, because after all, CO2 enters through the stomata, it's the only way it can get in. What if by opening up the stomata, you're going to lose too much water? So you now have to balance between transpiration and making food, going through the light independent reactions, the Calvin cycle. Plants have evolved in a mechanism around it, and that is a temporal, that is a time-dependent shift in when you absorb CO2. So what these plants will do is at nighttime, their stomata open up. The result is we can absorb CO2. The problem is we can't necessarily go through and use RUBP because we might have used it all up. So we add it to another acid. I don't remember which one it is. But we'll end up making an organic acid that during the daytime, we could then break the acid apart, release the CO2, and then run through the normal Calvin cycle. We're not worrying about photorespiration, adding a CO2 instead of, or adding an O2 instead of a CO2. What we worry about in this particular case is transpirational loss. This mechanism is referred to as crassulacean acid metabolism, or CAM. All it's attempting to do is bypass losing too much water in transpiration. The way it does it is it opens up the stomata, the guard cells swell up at nighttime, and they're closed during the day. So you can go through photosynthesis, but you don't need to worry about losing water. It's a very genius mechanism. When you want to know what plants do this, the one that I think of, here it shows you a pineapple, which is a monocot. It's in a very dry area. What I think of things like a cactus, which is actually the plant division of Crassulaceae. Or not division, that's a plant family. Is transpiration a big deal? Yes, because not all plants are as good at transpiration as others, meaning some lose a lot of water and some don't. This was an actual question, or a picture from a question. This was, I think, my second year teaching AP Bio. It's from 2011, and they asked you to calculate out transpiration. This is actually something that you're going to be asked to do in your lab. The catch is this one here was done in class. They had to create something called a pedometer. It's a major pain in the butt. If we had a block schedule, we'd spend the first hour making the pedometer so that the second hour we could do the lab. It's just a nightmare. It really is. We're going to be doing something far simpler. The catch is we don't get to make data that look like this. We get a before and an after and nothing in the during. But not all species will lose water the same way. We know this. Oh, I want to point out one other thing. And it's water loss here on the dependent axis. This thing here is written in a very odd way. It says milliliters of water lost per 100 grams of tissue. Well, why would you do that? Well, if species A turns out to be a 50-gram plant, species B turns out to be a 3-kilogram plant, it's really unfair to compare how much water they lose. So what you do is you level the playing field. We could say, well, here's how much water this plant loses per gram of the plant, or per kilogram of the plant, or per tissue, or 100 grams of the plant. It's a way of leveling it out. It's kind of like if you were to sit there and you go to the gym and it's, well, how much do you bench? Which is every question every guy asks. and you respond with your answer. And you can have a guy who weighs 100 pounds and he says, I bench 135 pounds. And you can have a guy who's 150 pounds and says, I bench 135 pounds. My question to you is, who's stronger? You'd say, oh, well, they both bench 135. Yeah, that's true. That's one way to view it. Or you could say, one benched 135 pounds per 100 pounds. The other person benched 135 pounds per 150 pounds. Once you view it that way, oh, I get a fair comparison, because now I'm comparing how much did you lift based upon your weight. And now we get a fairer comparison. It's kind of like, how many questions did you get right on the AP Bio test? Well, we could get answers, or we can talk about the answers based upon how many hours did you study, or what's your GPA, or when did you take biology? So is this your first time in the class, or second time in the class, or third time in the class? We can get different answers by having this per something. This ratio makes it so comparing numbers is fair, especially if I'm comparing things that are not the same. Species A and species B are not the same. So I need to figure out a way to level it out. And the way we do that is we take our value and we divide it by something that's somewhat in common between the two. Why would I point this out? Because not all plants are equal. I don't mean this to attack almonds. Almonds are delicious and I love almonds. They use up a lot of water. The question that we could ask is that because they're not particularly efficient at transpiration, or does it take a lot of water to make the almonds themselves? And what we find is they actually don't seem to be super great at transpiration. And it's not that they actually need a lot of water to make them, they're just not efficient when it comes to transpiration. And unfortunately, the Central Valley, where we grow a lot of almonds, it sucks up, it's rather dry. So it's not that conducive. That said, it's not the worst. There are plants that we grow that are far worse than almonds. But not all plants turn out to be the same. And almonds are actually far more efficient than drinking cow's milk or something like that. But that's meant for a baby cow and you're not a baby cow, so stop it. So tomorrow, you're going to be dealing with a lab that looks like this. Your task now is to hop onto the computers and you need to look this thing over. The reason why I say this is because you need to really think through what you want to do, because this lab is going to run for 2 weeks. This is going to be a 2-week long lab. You're going to set it up, and I know it says 1 week. I'm going to change it so it's going to run for 2 weeks. Because I've never done it for 2 weeks. I'm curious. We'll have a spot where, let's see if we can stop it after a week, but let's see how this goes. So maybe we'll test it for 1 week and then continue on for another week. What you realize is you can't just stop, so we'll have to do some that go for 1 week and some that will go for 2 weeks. But let's see how this goes. So this is going to be your task. As soon as I'm done, you'll need to be ready for tomorrow. Thanks.